Hi, I'm Ricky Milne. I'm a researcher in the Crop Immunity Group at CSIRO Agriculture and Food in Canberra. And today I'm going to tell you a bit about plant diseases, what can cause them, why we work on them, and how we're trying to help plants overcome them. Like humans and animals, plants also suffer from diseases. The group that I work in mainly focuses on diseases in cereals, so crops like wheat, barley, sorghum, and rice. Plant diseases can be caused by microbes, so things like bacteria, viruses, fungi, and so on. And if the plants lack immunity to these pathogens, something that everyone's probably pretty familiar with at the moment, the outcome can be devastating for the crops. And this can result in complete losses in the worst scenario. So this can have major impacts for not only the farmers, but for consumers and for food security. The main diseases we work on are caused by various species of fungi. I've got a couple of examples in this slide of diseases of wheat called leaf rust, stem rust, stripe rust, and powdery mildew. The various fungi that cause each of these diseases produce massive amounts of spores that can travel vast distances on the wind. They sometimes might be assisted by humans or animals until they find a compatible plant that they can infect, and they're then able to complete their life cycle. Whilst they might look pretty, they can have major impacts, and essentially what they do is siphon the nutrients from the plant, which normally would have gone into grain development, but instead they support fungal development, which then results in reduced yield for the plant and can sometimes actually end up killing the plant. I've got a couple of images here just to show how prolific these pathogens can be. And in this first picture here, we've got the stripe rust just taking over this whole field. And these other couple of pictures here where the tractors are going through the field, they're disturbing the rust spores and just resulting in these massive plumes that get blown around and can go and infect other plants. And so they can be very difficult to control. Unlike the current pandemic, we can't go around vaccinating plants. So one method that we can use to control rust diseases and other fungal diseases are chemicals like fungicides, although these aren't the most environmentally friendly control method. Using the plant's own immune system to control these diseases is a much preferred option over fungicides as it's more sustainable and environmentally friendly. There are a couple of different classes of plant resistance genes, and a nice analogy to think of them are these different types of door locks. So minor resistance genes, more like a privacy chain on the door, they confer a weaker resistance than major resistance genes, and sometimes this is essentially partial resistance, so they can slow the fungal growth down but not stop it completely. So in this example, you can see the susceptible leaf has lots of disease, whereas the resistant has some, but it's a lot less. Major resistance genes that are more like a deadlock on the door that stop it from being opened, they can confer much stronger resistance, and in this example, the resistant plant shows no fungal development at all and is completely immune. So we're interested in both of these types of resistance genes, and they can actually be used together. So minor resistance genes, whilst they do confer a weaker resistance, quite often they can last a much longer time in the field without being overcome than major resistance genes, and sometimes they can actually confer resistance to multiple different types of pathogens. Major resistance genes, while they do confer a much stronger resistance, they are prone to being overcome more quickly than the minor resistance genes, and quite often they can be very specific to a certain type of pathogen or even a certain strain of that pathogen. What we're doing to try and improve food security is to identify new and useful resistance genes, to understand how they work and develop markers for these genes to then enable the breeders to be able to incorporate the genes into their commercial cultivars for use against diseases in the future.